DNA libraries. Okay, so a genomic library is a collection of DNA fragments that are stored and propagated in a population of cloning vectors. Basically, you have a whole bunch of bacteria, and in each bacteria, you have certain segments of your DNA. Say the telomeric, or I won't even say that, the P arm of chromosome 1 happens to be in one bacteria. This allows research to study the entire genome of an organism. We're not going to watch the cute little video, but it is a nice little video on DNA library. I've given this to you in PDF format. You can take and copy it over. Now, new genetic varieties of animals and plants are being produced. And a plant with a new trait can be created using a TI plasmid. It's simply a type of plasmid. That's all there is to it. It's relatively easy to grow, and it's relatively easy to manipulate. So what I may do, I take this TI plasmid, I cut it open, I take and I cut around the desired gene, and then I take this plasmid, I introduce it into my plant cell, and now I have a plant with a new trait. Perhaps it is some sort of pesticide resistance. Perhaps it is some sort of herbicide resistant gene, something along those lines. You can do some really amazing things with this. Golden rice actually contains beta carotene in it. That's what's found in carrots. And it helps prevent vitamin A deficiencies in populations that do not have access to a lot of foods with vitamin A in it. We in America, we put stuff in even just our basic white bread to help prevent these nutrient deficiencies, but a lot of third world countries do not necessarily have access to this type of food. So this is a way to help prevent that. Vitamin A, by the way, is very important for night vision. You can have drought-resistant corn. The Flavor Saver tomato, which is a Cal gene, as I recall, that's Caltech is what created that one. And these are just, they're really good tasting tomatoes, purportedly. I don't eat just plain tomatoes. I prefer it in sauce. Drought-resistant corn can be hugely beneficial when you're trying to grow corn in a region that isn't as wet, or even the American Midwest that it's a prairie. You have very huge amounts of fluctuation in the weather from year to year. Some years are really, really dry. If we can have drought-resistant corn, it means that we aren't having a major hit to those sorts of crops. Transgenic organisms. These are organisms that have had genes from other organisms inserted into them. Different organisms, including bacteria, yeast, and mammals can be used for this purpose. For example, these cute little sheep here actually carry a gene for human blood protein that is potential treatment for cystic fibrosis. There are cows that actually have been genetically engineered that their milk contains in it clotting factor 9, which is what is mutated in people with hemophilia. Hemophilia is a disease where your blood cannot clot properly. We talked about it. It was the whole Russian Tsar family situation going on. And instead of these kids having to have injections frequently of this protein, they can drink milk instead, which, face it, is sort of nice. Now, before I go on to the next thing, GMOs, there are a lot of pros and cons to GMOs. This is another one that gets published a lot in the media. This is another one that could, at least in my case, lead to potential family debates at family gatherings. I personally, yes, there are things I'm concerned about with GMOs. I don't like the concept of Monsanto being able to just dump as many herbicides as it wants to on top of my food, because I've now made it that my corn will not die with it, but all the weeds around it will, just because are that many herbicides or that many pesticides really good for my general consumption? But as far as safety, just because it's been genetically modified, that I'm not so concerned about. We've been doing that for literally millennia as soon as we started figuring out we could breed certain things together. DNA technology is changing the pharmaceutical industry. It's widely used to produce medicines and to diagnose diseases. Hormones, being able to create hormones, whether it be human growth hormone, whether it be insulin, whether it be estrogen and progesterone to help combat symptoms of menopause, whether it be testosterone to help. We're finding that men, as they age, their testosterone levels can fluctuate, that can cause more dramatic health problems than erectile dysfunction. Not that that isn't a problem for people, and I don't want to downplay that, but there are other health complications that can come with fluctuating testosterone levels that this can help. Cancer fighting drugs. Being able to look at the genetic markers in cancer and be able to create specific chemotherapies that can fight that. For example, Mrs. Coos' sister-in-law had breast cancer. They biopsied, they grew the cancer cells in petri dishes, they separated it into 3 groups, and they actually exposed the cells to 3 different chemotherapy drugs, figured out which one worked the fastest, and that was the chemotherapy they gave her. That's huge to be able to use this sort of technology to really fine-tune people's cancer treatment. New vaccines are being produced with DNA technology. This particular machine is being used for a vaccine against Hep B. Hepatitis was just beginning to be a vaccine when I was in high school. I did get it in high school. Meningitis did not come out until I was in college, and all of our parents made us go get it. I had a classmate that actually died our freshman year of college because of blood meningitis that was unfortunately misdiagnosed in the emergency room. But your Gardasil, you guys haven't had the chicken pox. You've been vaccinated against that. The shingles vaccine, all of these are things that are being able to be developed with this new technology. Human insulin is actually produced by bacteria. We used to use pig insulin, actually, which, I mean, yes, you can get it from pigs. It's not quite as effective, but it was relatively effective. Downfall, pigs are a lot more expensive and a lot dirtier to maintain, whereas bacteria, you can literally produce thousands of gallons of this a year in relatively simple conditions. And it dramatically dropped the cost of insulin, although I know insulin is still redonkulously expensive, but it actually did help improve it. Okay, DNA technology is being used increasingly to diagnose disease. Some of it is you're looking at genetic disorders. We're finding more and more the genetic component of high blood pressure, high cholesterol, those things that when you get old like me, you go to your doctor and you worry about diabetes, things like that. We're trying to get it so that we can really begin to diagnose disease. With what we have going on right now with COVID-19, they are trying to create assays that will diagnose more quickly. When it first came out, it was 3 to 5 days to get a result. Now it's about 24 hours to get a result. They're really hoping to get it down to less than that. The fact is, when I was a child, it took 24 hours at least to get a positive strep diagnosis. Now you literally walk into the doctor's office, they swab your kid's throat, 15 minutes, 20 minutes later, they come back, they say, yep, your kid has strep. Okay, influenza is the same way. At least your major strains of it, diagnosed within 20 minutes. That's what we're trying to get to with COVID-19, okay? We're also then trying to look at antibody assays to see who has been exposed, who has antibodies against this. Now, we still don't know whether or not these antibodies mean that we cannot become reinfected with COVID-19. We have not had a long enough period of time to be able to really study this and see the results of that. But there, even on the news the other day, they were talking about they did a sample of 3,000 people or something in the greater New York City area and found that 14%, I think it was 14% of the population had the antibodies, which means that they have already been exposed to the disease, already had it, already gotten over it. So that's pretty amazing. And then vaccines, helping research to develop vaccines. Again, something that's happening right now with COVID-19. We are trying to develop a vaccine against it because truthfully and honestly, it's not going to go away unless we vaccinate. That's just how viri work. I'll stay off of what I think of anti-vaxxers. If you ever want a real fun time, you ask me about that. And we will stop there until our next one.